The technology-driven innovative teaching learning method. The students have different style of learning. They may be visual learner, auditory learner, kinesthetic learners, or learners by reading and writing. Most of the students are a combination of these four styles, and we have to address to these learning styles when we teach during our sessions. The interactive teaching not only uses the blackboard teaching but also the PowerPoint presentation. Both the modes are blended in together to bring out active engagement of the students. The writing tablet can be used as an innovative teaching tool. It can be used in an interactive lecture just like a traditional blackboard, but only it is more versatile. Hand-drawn images, graphics, and diagrams can be used to create information-rich content for interactive learning atmosphere. It can be used in large group settings, the small group setting, and even for online teaching methodologies. Kahoot is a useful gaming tool that can be used by a teacher for the assessment purpose. The students can download the Kahoot and then the teacher can create the quiz, and on entering the game pin for the Kahoot, the students will be taken to the quiz. Once the teacher starts with the quiz, the students are guided into the quiz and then they can see the questions projected on their screen, as you can see on the left. So once the questions are projected, the student can attempt the questions within a given time frame, and then the correct answers which are there for the quiz will also be displayed simultaneously. This can be used as a self-assessment tool by the students and also by the teacher to know where the students are lacking in their knowledge. Google Form can be used for the purpose of assessment and more importantly also for the feedback. So our learning today will be on the topic of synapse. You have heard of this disorder? Schizophrenia, bipolar disease? Yes? Yes, they are the neurodevelopmental disorders. Or you have this disorder which is Parkinsonism, which is again a neurodegenerative disease. So what's the role of synapse in this is that whenever there is a structural or a functional dysfunction in a synapse, it can either cause these symptoms, cause this disease, or it can be a consequences of this disease. So what do you learn at the end of this session? It will be the structural and functional aspect of the synapse, as you correctly said in the chat box. You'll be learning to define a synapse, enumerate the different types of synapse, and then regarding the function of the synapse, you'll be studying about the transmission across the synapse. So this is for the recall. We have already studied this. It is a neuron. You can type in the chat box the structures you can identify as 1, 2 and 3. Okay, yes, okay. So structure 1 is the cell body, 2 are the dendrites, and the axon is extending from the cell body to the nerve terminal, and that is structure 3. Very correctly said. The word synapse is taken from the Greek word which means a junction, and since we know that there is no anatomical connection or the continuity between the different neurons, they are functionally connected. Now, depending upon the different types of anatomical connections between the two neurons, they can be exodendritic, means that axon of one neuron will form a synapse with the dendrite of the another neuron. Then we have the exosomatic, axon of one synapsing with the soma or the body of the another neuron. And then we have this exoaxonic, which is the synapse between the axon of a neuron with the axon of another neuron. This is very less common type of synapse. The physiological classification of the synapse can be the electrical synapse, the chemical synapse, or even the conjoined synapse. First, talking about the electrical synapse, they are nothing but the transmission of a nerve impulse through these gap junction, and we know that these gap junction are connecting the two neurons, so that allows the easy transmission or the fast transmission of the nerve impulse. So this type of conduction is also known by the name of electrotonic conduction, and this type is seen in cases of one cardiac muscle to the another cardiac muscle. So they are basically connected by these clusters of ion channels, and therefore they are bidirectional in nature. This is seen in retina or even the olfactory bulb, and the connecting unit here is the connexon. So this type of electrical synapse offers high speed conduction to the neuron. Now this is the chemical synapse, you can see, and it is the most common type of synapse which is seen in the central nervous system. So this, the neurotransmitter being the agent that carries the nerve impulse. So the information travels only in one direction, and they are fatigued, they can be fatigued. So this type of nerve synapse is present in type 1 and type 2 category. So we'll understand about this synapse. The conjoined synapse refers to a synapse where both chemical and the neural electrical type of transmission is seen. It is seen most commonly in the vestibular apparatus. It has speed as well as is the transmission by the neurotransmitters. Now coming back to the chemical synapse, which I said is the most commonest type of synapse. First, let us understand the structure of this chemical synapse. These are the different parts, that is synaptic knob, the presynaptic membrane, the synaptic cleft, and the postsynaptic membrane. With the help of this, you can see these axons which lose the myelin sheath near its ending. When they branch, they form this swelling. The swelling of the neuron at the axon terminal is known by the name of, what do we call it? We call it, here we can call it synaptic knob. This synaptic knob or the terminal button is a part of a presynaptic membrane. And here we have another neuron which is forming a synapse with this presynaptic terminal, and this is known by the name of postsynaptic membrane. Okay, so we have presynaptic membrane and postsynaptic membrane, and this may be a dendrite and this might be a axon. So the type of synapse which will be formed will be exodendritic. And you can see this gap between the two neurons. Okay, so this is known by the name of synaptic cleft. Okay, so we have these three structures: the presynaptic membrane, the postsynaptic membrane, and in between we have these synaptic cleft. So if I talk about this presynaptic membrane, I have this structure which is a voltage gated channel which is present here, which is very important in the transmission of nerve impulse. And this is what? This is L type of voltage gated channel, and which ion will flow through it? It will be calcium ion. Okay, so the calcium influx is by this voltage gated channel. So beside this voltage gated channel, we have some other structures. One is synaptic vesicles. The neurotransmitter is stored in this synaptic vesicle in these densely packed structures which are small in size, and they store mainly the catecholamines. Okay, and when we talk about the larger dense particles, dense neurotransmitters being stored in these vesicles,
it is mainly the neuropeptide. So you see the difference? It is the catecholamines here, and here we have neuropeptide. And then we have small clear vesicles, and they store a neurotransmitter which is acetylcholine. So we have different type of neurotransmitters which are stored in a different manner according to the amount of neurotransmitter which is present. And one of the important structure that we have in this is this. And which is this structure? It is mitochondria. Because mitochondria will be responsible for providing energy for the synthesis of neurotransmitters. And then we have these, which are microtubules. They facilitate the movement of these synaptic vesicles towards the nerve endings. Okay. And here we come to the synaptic cleft. And I said it is a gap, maybe 20 to 40 nanometer between the two neurons, and it is filled with extracellular fluid and glycoprotein, and these neurotransmitters are released in this gap. Okay. And then we have this postsynaptic membrane. Postsynaptic membrane में क्या होगा? The postsynaptic membrane will have the receptors. So this serpentine receptors which is present here, it is G protein coupled receptor, and it is a type of metabotrophic receptor, okay, which is linked to the G protein, linked to the enzyme system, which is responsible for the influx of ions. Or your second type is that which is responsible for the influx of ions directly. The ions may be sodium, potassium, or chloride, and this is known by the name of ionotropic receptors. Okay, so we are clear with the structure. We have the presynaptic terminal, the postsynaptic terminal, and then we have the synaptic cleft. So now we can talk about the events which are responsible for the generation of neuro transmission across the synapse. So first, whenever this event is there, first there is transmission of nerve impulse that is coming from the nerve ending, and then it reaches the nerve terminal. This is the first step where the impulse that is present in the nerve fiber reaches the nerve ending. Then second step will be the opening of this channel, and this channel is the voltage gated calcium channel, which will lead to the influx of calcium ion. So one calcium ion is inside. It will facilitate the movement of these synaptic vesicles, and these synaptic vesicles will then find a place in the membrane, and they will cause the release of neurotransmitters here. So one important point about this is once they release the neurotransmitter, they get sealed. So this process is known by the name of kiss and run discharge. Okay. So once there is release of neurotransmitter, the membrane gets sealed in that manner. So the process of exocytosis is mediated by the calcium ion. Okay. So once the neurotransmitter has reached here, now what will happen? What will happen? So what will happen? Either this neurotransmitter will be taken up. It will be reuptake of neurotransmitter. Either it will be degraded by the enzyme, or the third most common action will be, it will be acting on these receptors here. Okay. So the receptors, it will be acting here. So it can have two type of action. So what are these two type of action? First is it can cause the generation of positive type of positive type of ion movement that will lead to excitatory postsynaptic potential, or it can lead to the negative potential, which is known by the name of IPSP, which means inhibitory postsynaptic potential. Okay. So finally, these will sum up and decide whether the nerve will be excited or it will be inhibited. So when we talk about the event, so we have the first event, that is this event, that is traveling of action potential. The second one is influx of calcium ion. Third is the movement of neurotransmitter, the synaptic vesicle. Fourth is the presence in the synaptic cleft. Fifth is the action on the receptors which are present. Okay, so we can further see this across steps of transmission across the chemical synapse. The first one will be the nerve impulse reaches the presynaptic terminal. The action potential is reaching. You can see in the figure. The second one is opening of the voltage gated calcium channel and increase in ion, that is calcium ion. The third is, as I said, movement of these vesicles, and these vesicles will then discharge in the.
Now let us evaluate our learning till now. So I'll be clicking on this link and you have already downloaded Kahoot app. So you'll be getting a gaming pin. Enter that. You'll enter into a quiz and then try to attempt that. These are the links for the additional resources, the pre-reading material, and even YouTube link for the lecture. The follow-up assignment you can submit in the next session. It is a crossword. Solve it and then you can submit. The content of the class has been taken from these reference material. My sincere thanks goes to Principal Sir of Dr. YSPGMC Nahan for providing permission for this study, the faculty and staff members of Department of Physiology and students of Phase 1 MBBS for participating in the study. Thank you all for your patience.